Uh, Good afternoon. So now today we discuss the another topic, that is the genome organization in higher plants. So yesterday we discussed that one, the genome. What is the genome? And so genomics, you know that one. Study about the genomes, known as genomics. So the genome, that is the DNA particular in all organelles, means microorganelles as well as plant macroorganelles. That one, that one we call as a genome. So, what is the gene organization in the higher plants? Once you see that one, yes. So, in the small number of plant genomes have been studied in the great detail in 19th century, early in the 21st century, and DNA, the genome has been sequenced. So here I shown to the diagram. There is the Arabidopsis genome.
Uh, this is the functional classification of Arabidopsis genome. See here,
Eukaryotic model organism. So that one is the Arabidopsis.
one of the first eukaryotic organisms that will be completely sequenced in the small.
Species, that is the Arabidopsis salina. During the past decade, that is the Arabidopsis has emerged as one of the most widely used model organisms for studying the genome in higher plants. It was chosen for the sequencing because it is a highly compact genome of about 130 megabytes with a little interpresent interpreted repeat two.
So see here, that is the
All are there. So studying the plant genome, studying the plant genome are more complex than other eukaryotic genomes. And analysis that reveals many evolutionary slips and tons of DNA sequences over time. The plant molecular genome ranges in size from less than 100 million base pairs to more than 100 million base pairs. The plant shows widely different chromosome numbers and varied polyad level. Overall, the size of plant genome, both number of chromosomes and total nucleotide base pair, exhibits the greatest variation of any kind, any kingdom in the biological world. So, for example, in the tulip plants containing over the 170 times as much DNA as a smaller breed of the Arabidopsis. The DNA plants like animal can also contain the regions of sequences. Repeat the sequence inversions and transferable element inversions, which further modify their genetic material. The traditionally and polyploid has been used how
This pattern
evolved. So increasingly researchers are turning to study in the organization of plant DNA sequences to obtain important information about the evolutionary history of plant species. Another here I shown to the haploids and polyploids. So wait, the picture. What is
plates, diploids, and polyplates also. The diagram shown here. See here. The chromosome number is possible in plant genomes. Sometimes here haploid. This one is the total haploids are there. The haploids, the diploids. These diploid character is present. So here this one is the more than two chromosomes are there. That condition we call is a polyploid. So haploid variation, diploid, and polyploid condition. The nuclear genome of rice consists of 450 million bases of DNA divided among the 12 chromosomes. Base pairs and include the genes that encode some 38,000 proteins are there. Along with their controlling these sequences, the genes is represent less than 10% of the total amount of DNA, and half of the all the DNA consists of repetitive motif to that is the represent thousands of times in the genome. So another fully sequenced plant genome that is Arabidopsis has total 157 million base pairs with the 31,000 genes on 5 chromosome pairs. So it is currently anticipated that the complete genome sequence of Arabidopsis will be at the end of the year 2000 here, because the Arabidopsis is only distantly related to cereal crops, which is the provide the bulk of the world food supply. That the genome of rice will be also sequenced and during the next decade. The rice was chosen because in additionally it is the importance as the food source for about the non-quadrant of the human population. It has one of the most compact genome among the cereals. It contains about 3.5 times as much DNA as Arabidopsis, but only about 20 percentage as much DNA as maize, and about 3% as much DNA as wheat.
So, in the all the higher plants at the diploid level were approximately
And regulatory
in a sequence for physiological process, seed germination, growth, flowering, and reproduction. For the nuclear genome sizes measured by the number of base pairs of DNA of different plants.
in our thick and relatively constant genome size, the amount of nuclear DNA can be given as an absolute weight of the DNA, or they converted into the number of base pairs represented by that weight. So I'm shown to the some different plants here. So different types of the plants, and their genome sizes means million of.
Yes, next I show here. Wait.
This is the base pairs of different Arabiopsis and plumas, citrus, oryza, petunia, and Pisum, Avena, Tulipa. So these are the some plant species which is the so the common name is there, botanical scientific name and the common name, and another one the genome size mbps. So million of base pairs, how much of base pairs are there? Okay. So these are the around here what happened here. Just I explain. So Arabidopsis here commonly Arabidopsis here around 145 million base pairs are there. So Prunus, that is the 262 base pairs are there. Ricinus caminus, that is the castor beans, we call as the castor. So 323 base pairs are there. Citrus, another one is the citrus. So that commonly we call oranges. So these orange have the 367 million of base pairs of genome size, which is present in the citrus. Another one is the Oryza sativa, that is commonly rice. In rice, here 424 base pairs are present, million of base pairs. So remaining petunia and pseudium, that is the Pisum sativum, yes, garden pea plant. So Pisum sativum here 3,947 million base pairs which is present in the Pisum. The tulip,
Garden tulip, 4,000.
million base pairs are present. Okay.
So, So plant nuclear composition. So that is the plant nuclear composition means the genome consists of DNA divide among the chromosome within the cell nuclear. So the plant nuclear genome, which is DNA, which is the chromosome with the cell nucleus. The plant genome contains coding and regulatory sequences for the genes and repetitive DNA. The genomes are evolutionary and insights into the evolution of genes, sequence, families, and genome, and support the study of specific phylogeny and the relationship means the phylogeny relation. The plant nuclear genome consists of DNA and it is contained with the nucleus and organelles that is encapsulated by the double membrane in each cell. During the cell division, mitosis, the genome condenses into a characteristic number of metaphase chromosomes and nuclear membrane breakdown and the chromosomes divide moving into the two daughter cells before the nuclei reforms. So there is a the organization of plant genome. What is the here organization of plant genome? The most seeded plants, the containing quantities of DNA that greatly exceed their need for the coding and regulatory function. So hence, for the plants and very small percentage of the genome, which is actually encoded the genes involved in the production of proteins. You know that one. So each means DNA code, they have synthesis individual proteins. Individual proteins are synthesized and this proteins of genome which is encodes most of the transcript genes is often is referred as a low copy number of DNA because the DNA sequences copying these genes are present in the single or small number of copies. The medium copy number DNA is composed largely of DNA sequence that encode ribosomal RNA or RNA, that one we call as the rRNA. A key element of the cellular machinery that is the translate, transcript, the messenger RNA, that one we call as mRNA, into then synthesis of protein. So DNA then encoded into the rRNA means ribosomal RNA. That ribosomal RNA then synthesis the mRNA means messenger RNA. After mRNA, they are to synthesize the proteins. In plant genomes or RNA genomes, maybe repeated the several hundred to several thousands times. The plant cell may also contain the exceed DNA in their genomes in the form of higher repetitive sequences or high copy number of DNA. At present, the function of this higher copy number of DNA in plant genome, sometimes which is the unknown condition. Okay, and sequence of replication and inversion. The high copy number of DNA sequence in the plant genome may be short, such as the nucleotide sequence and GAA, are much longer, involve up to several hundred nucleotides. Moreover, the number of copies of an individual high copy repetitive DNA sequence can be total from 10,000 to 1 lakh. There are the several possibilities for how high copy repetitive DNA sequences may be organized within a plant genome. So several copies of a single repetitive DNA sequences, which is may be present together in the same orientation in a pattern called as a simple uh, tandem array. Alternative, that is the repetitive DNA sequence, can be dispersed among the single copy of DNA in the same orientation, that is the repeat or single copy interpretation of the opposite orientation, that one we call as the inverted repeats. In addition, group of repetitive DNA sequence can also occur together in the plant genome in a variety of possible arrangements, such as the compound tandem array or the repetitive interpretation. The presence of repetitive DNA can vastly increase in the size of plant genome, making it difficult to find and characterize individual single copy genes. A variety of mechanisms can counted for the presence of highly repetitive DNA sequences in the plant genome. So, what is transposable elements? Here, some elements, that is the transposable elements, are special sequence of DNA with the ability to move from place to place in the genome. So that is easily to move from one place to means another place. That is the this one called as jumping genes also. They can exercise from one side to unpredictable times and reinsert in other site. This is the reason is the transferable element, that one we call as a jumping genes also. Transferable elements are jumping genes. So these transferable elements,
Coding regions or regulatory regions of gene, and so affect expression of that gene, resulting in a mutation that may be not be detectable. So here,
Barbara McClinton is own these identify these jumping genes in 1983 are transferable elements in the corn. Okay, so in the corn, these McClinton is identify these jumping genes. So I shown to that is the how they are to shifted to one place to another place.
So I shown to here that is the transferable genes or jumping genes. So that is the simple tandem array. That is the repeat or single copy interpretations.
So that is the transfer gene shifting to the single copy gene. Okay. This is the so another is the chloroplast genome. Plast genome or its evaluation. The chloroplast is a plant organelle that function in the photosynthesis and it can independently replicate in the chloroplast is a very important cell organelle which is covered the double layer cell organelle and involved in the synthesis of carbohydrates means involved in the photosynthesis so called as the independently replication means they have the DNA there. That's why the plant chloroplasts have their own specific DNA, which is the separated from the present in the nucleus. So these chloroplast DNA, they have own DNA, which is not in the part of the nucleus. This DNA is material inherited and encodes unique chloroplast proteins. Many of the proteins encoded by the chloroplast DNA are involved in the photosynthesis. So this one means the encode
DNA coda. They are the involved in the synthesis of their proteins.
plus DNA is the chloroplast DNA is present as a circular loops of double-stranded DNA, similar to prokaryotic chromosomal DNA. They're only the
That is up here. Which is similar to prokaryotic chromosomes. The DNA.
bacteria. So the chloroplast DNA contains genes
That are very similar to those present in the prokaryotes. This character is also similar to the bacterial structure. The DNA in chloroplast of all plants, that is the same number of genes and they are the present in about the same order also. So I shown to
Mm, right.
so this is the chloroplast genome. Chloroplast genome. Here the small single copy region.
The inverted repeat large single copy.
So this is the mapping of genome. Is the gene mapping is widely applicable approach to scanning and genetic information.
An organism for genes are responsible for a specific traits. Yes. Higher plants are thought to have 25,000 or more genes, majority of which is remain unknown function. A particular strength of genome mapping is the facilitates isolation of gene bases simply on the measurement of their effects on phenotype, requiring no a priori knowledge of the biochemical function of performed by the gene. So much more of genome can be mapped using the RF uh, fragment length of polymorphism. So which is need not to have the macroscopic phenotype.
Or the pattern.
DNA fragment produced when the DNA is treated with the restriction enzymes that cleave at a specific sites. RFLP mapping that can be identified important region of the genome at the glance. The RFLP mapping can be identify the important region of the genome at a glance, while the sequence data is require sophisticated computer-based searching and matching the system. So a comparison of RFLP maps of parent and progeny, which can be indication of the heritability of gene traits and of heritable loci that are the characteristic of traits. So another tool that is the utilize sequence variability of AFLPs, that one we call as amplified fragment length polymorphisms. So hybriding the DNA primes which with the genome DNA fragments that have been cut with the restriction enzymes that E. coli or MCL and subsequently amplified using the polymer PCR polymerase chain reaction generated AFLPS maps, resulting the PCR production, which is represent each piece of DNA cut by a restriction enzyme or separated by size via gel electrophoresis. The band sizes of ALPS gel tend to show more polymorphism than those found with the RFLP mapping because the entire gene is visible on the gel. So, what is the applications? Applications. The genomics research will be lead to a new innovative ways to achieve the trait importance and clearly genomic can be helped to support the related food safety and food quality and food diversity. The genomics provides a objectively in the breeding as never before possible. It allows hypothesis testing of quantitative genetics applications in plant improvement. It is also useful to increase the crop productivity, improve crop quality, and maintain the environment. Okay, so this one is the genome organization. So,
Another topic here, structure and
Mahesh, eukaryotic genes. On the so, wait, few minutes, we wait. So here what is the here structure and organization of eukaryotic genes? Here, now we explain the
So explain here.
in the eukaryotic gene structure and organization. See here the diagram, the central dogma here DNA.
Mm,
This mRNA, that is the transcription, that mRNA then synthesis the rRNA and rRNA then synthesis the proteins in the process of translation. Okay. So in the eukaryotic transcription complex, here I show to which there is the in the eukaryotic transcription complex. Here what happens? See here the exons. So expressed to the coding sequence that is the transcript and translated. An exon is the portion of gene that code of which is have the amino acids. A gene starts and ends with the exons, that is 5 to 3. So exons include the both untranslated region and the protein coding translated genes. So introns means the intervening, that is a non-coding sequence. They are removed by the process of uh, splicing. See here, that is I shown to uh, introns and extrons. Yes, okay. This one is from, see the diagram. Extron, exons and introns, it is there, and these introns to develop as a a mRNA by the dressings, clippings. So promoter sequence.
That is the transcriptional initial region, the region of RNA polymerase begins, which is located directly upstream at the five ends. The RNA polymerase and the transcription factor bind to promote the sequence. They define the directed, the indirect with the DNA transcript. So another after termination sequence, here to the define the end of the transcriptional units such as the gene and initiate the process of releasing the newly synthesized RNA from the transcription machinery. So enhancer means the enhanced transcription of associated gene when they bound to transcriptional factor. Enhanced sequence can be positioned in both favor and revert sequence orientation. So silencer, what is the here silencer? The DNA sequence capable of binding transcription regulation factors called as a repressor. So repressors are there. The silencer is preventing the gene from being the repressor proteins.
So another one is upstream and downstream is also there. That is the synthesis of DNA and next one we discuss the chloroplast and genome. So chloroplast and mitochondrial genome. See here, wait few minutes. So in the so next one is the chloroplast and mitochondrial genome structure and function. We have to the mitochondrial resemble bacteria. So how we just I show to the figure. of you.
Sure.
So, here that is the mitochondria is there.
Chloroplast, Golgi body, and the nucleus is present. Two popular theories presupposing the naturalism keep to explain the origin are membrane-bound organelles. Two membrane-bound organelles. Endosymbiosis, invagination. These two theories are there to explain the origin of mitochondria and chloroplast by the in 1981. So another one is the invagination here, the plasma membrane to form the endomembrane system. The most general biologist text bacteria, how they are too similar to the bacteria. The mitochondria resemble bacteria in size and morphology also.
Are bounded by a double membrane. Mitochondria. No. Organelle. The outer through the derived from the engulfing vesicle and inner from the bacterial plasma membrane. Some enzymes and inner membrane transport system resemble to prokaryotic plasma membrane system. The mitochondrial division resemble to the bacterial binary fission. Similarly, here the bacterial division is also that is a binary fission. They contain the small circular loop of genetic material that is the DNA, which is also similar to the bacterial DNA. They produce a small number of protein using their own ribosomes, which look like a bacterial ribosomes. So that's why the ribosomal RNA resembles to the eubacterial RNA. So these are the some similarities to the bacteria and how they do not resemble to the bacteria. The mitochondria are not always size and morphology of the bacteria. Some trypanosomes. So the mitochondria undergo the spectra search that do not resemble bacteria during the different life cycle stages. It's discovered by the 1971.
In the proteins, the mitochondrial division and distribution of mitochondria, daughter cells, is tightly controlled by even the simplest eukaryotic cell. So now the mitochondria. Few minutes.
Mitochondria are the organelle found in most organisms. The site of the Krebs cycle and electron transport energy producing process during the aerobic respiration. These are occurred in the mitochondria. Are inherited only from the mother during sexual reproduction in mammals, are probably all other vertebrates. Because of their mode of inheritance, genetic material found in the mitochondria appears to be useful in determining the maternal linkage of organisms. So see here, this is the mitochondria structure I shown through the figure.
Uh, so this is the this is the structure of mitochondria. The matrix, the outer membrane and inner membrane, and some cisternae folding areas are there. That one we call as a cisternae. Okay. The extranuclear DNA, the mitochondria and chloroplast have their own DNA.
Extranuclear
Inheritance. The recombination is known between the some uh, matrix and ctDNAs. The extranuclear DNA may also be called as the cytoplasmic DNA. Generally, that is the mitochondria means matrix, and the cytoplasmic DNA is circular as containing gene for the multi protein. Some portion of which is also coded for the nucleus. The extranuclear DNA has their
mutations is independent of nuclear DNA.
but not always all the
Uh,
transcription and translation are found in mitochondria or the cytoplasmic DNA. Only that.
So mitochondrial DNA is generally small in the animal cells.
Can be more than the uh,
Of the magnitude larger, the plant mitochondria
size.
and content with the large Arabidopsis mitochondrial DNA is around. Seven in the Cyclomonans mitochondria DNA of 96. Most of the genetic specification for the mitochondrial and function is residues in the nuclear genome with the important into the organelles of proteins and yes. Uh, so development of this one is the mitochondrial DNA. Okay. Now coming to the chloroplast. So development of chloroplast, that is the etioplast, amyloplast, and ila- ilioplasts. Etioplasts, there are the intermediate type of plasts developed from the protoplasts that have been not been exposed to light. So amyloplast, another one. See here, I am shown to the diagram. What is the diagram? Here I show.
The element of chloroplast. Here, different types of the plastids are present.
See here. The different types of plastid, etioplast and protoplast and chromoplast, chloroplast, leucoplast, amyloplast, leoplast, proteinoplast, and talolith, statoliths. So these are there. So and
Amyloplast. That one is amyloplast because the converts glucose into the starch and stored starch grains in the stroma. These stomata, that is the amyloplast. And another one, ileoplast, synthesis and storage of fatty acids and terpenes and other lipids. So, origin and evolution of chloroplast contains 3000 proteins. The present day, the chloroplast genome contain only about the 70 genes are present.
nucleus.
Are really similar to those in the cyanobacterium.
The rapid and massive reduction in number of genes transferred to nucleus lost. 80 to 90% of plastic proteins are encoded in the nucleus. A great overlap in the gene content suggests that the last common ancestor of DNA had that is there. Another one is the inverted repeat. The ranges from 5 kilo kilobytes to 76 kilobytes in the length, and that contains RNA genes plus others, that is the on in the brown algae, and 10 in the tobacco and geranium. So present in the land plants and chlorophytes, chromophytes, partial in the coniferals. Another one is coding region. Four ribosomal RNA genes, which is present at the 30 tRNA genes, more than 72 genes encoding polypeptides, several converted reading frames coding for the proteins of at unknown function. The plasmid genes coding for the polypeptide can be classified into several categories. Here, the genes coding for the prokaryotic RNA polymerase, core enzymes, the genes coding for the proteins of the translation aperture, for the photosynthesis aperture, genes encoding the subunit of the DNAH, that is the dehydrogenase. So plasmid gene proteins, that is the it is the properties of this gene plasmid genes. No plasmid tRNA gene code for the its three-strand chloroplast, and no RNA, even the small size is imported into the chloroplast. The plasmid genes of higher plants containing the single introns. The introns have been classified into two groups, so that is the group 1 and group 2. So another one is the chloroplast division. Many of the bryophytes and pteridophytes possess the one single plastid in a single cell. For example, Isaitis, an evaluated fern, possess one chloroplast for there is a meristematic cell and several chloroplasts in mature cells. The cell in the angiosperms, the dark green spinach leaves containing the more than 200 chloroplasts per mesophyll cells. Either that is the Arabidopsis containing more than 100 plasts per mesophyll cells. So chloroplast means the plastid division control. When the number of plastids per unit in the low, the plastid division is probably controlled by the cell cycle only. When they contain the large number of plastids, they do not divide synchronously. The regulatory pathway that is determined when the plastids enter the division cycle is unknown. So, and recently discoveries that the division of plastids, besides that overall control by the cell, which is prokaryotic like mechanisms. So, replication of plastid DNA. All the plastid chromosomes is a circulated cell of the tobacco replicate in one cell cycle. The DNA synthesis occur outside of compact nucleus. Uh, so mechanism that is the governing the replication of plastid. the forming two displacement lobes. I show to here diagram. Wait. So this is the
Mechanism replication in the plastic DNA. So loops, that is the D loops, see here, and Crane's type of
so the plastic DNA contain also rolling the circular replicative intermediate. So enzymes participating in the replication. So DNA polymerase resistance to amphidicholine and inhibited by the ethidium bromide and 43 kilodalton protein, 120 kilodalton primer. So two different topoisomerases. The topoisomerases two active have been detected in the chloroplast of higher plants, and there is the grace activity. DNA helicase is there. So transcriptional operates. So that is the regulation of chloroplast. Chloroplast gene is expression after level polymerase. The sigma-like are controlling the area of the plastid encoded plastid RNA polymerase transcription fact is the two types of RNA polymerase and the regulate the size of transcriptional system. Plastid subunits uh, will be and this is related to polypeptide polymer.
The transcription initiation of factors. That is the translated product of six different cDNAs shows the strong similarities with the prokaryotic sigma 70 factor. The regulation of relation involve the power relation and sigma like factor. Inter- parts of the plant factor have been different functions. That is the N-terminal part of the sigma 70 factor. From the E. coli. The C-terminal part, which is responsible for the DNA promoter, recognized in the functional conserve between the prokaryotes and plastids. So, the nuclear encoded plastid RNA polymerase, the resembling the bacteriophages G7 RNA polymerase, and nuclear encoded plastid RNA polymerase transcribes the genes encoding the elements of the genetic system rather than the photosynthesis genes. Hypothesis attributes specific function to NEP means nuclear encoded plastid in the housekeeping genes expression during the early phase of the plant and plastid development. So this is the thank you so much for the genome organization structure in the higher plants, how the genome is function and application. And another one is the discuss here mitochondrial. Uh, Mitochondrial gene and chloroplast genome, then replication and what is the main structure and functions of the mitochondrial DNA and as well as so okay. Thank you so much for attending the classes. Thank you so much.
So thank you everybody. And the tomorrow and another class we have to discuss another lesson. Thank you everybody. Thank you so much.